I'm a genetic pathologist, and in case you're not aware of what genetic pathologists are, we run the diagnostic laboratories that apply genetic tests to medical diagnosis. And while I share Chris's passion for great discoveries he makes in mice, I have to explain the purpose is not to extend the lifespan in mice, but rather to apply and extend the lifespan in people. And that's my role, to take the testing and to introduce it to the diagnostic laboratory. But let me give you a little bit of an insight as to how medicine actually works and where the pathologist fits into the system. So when you're not feeling well, you may spend time in bed, may consult a doctor, and inside the doctor's mind, after they've been listening to you, if they're patient, they'll listen to more of you, less of you, they'll start forming hypotheses as to what your aches and pains might be all about. And then from this, they'll come up with some thoughts as to what particular diseases or conditions may be affecting you at the time. And from that process, they will move forward and try and work out what is really the cause of what's happening to you. Patients don't present with diagnoses, they don't present with diseases, they present with aches and pains and symptoms and signs. And the doctor will come up with a thought and then we'll request a test. And you've all had pathology tests, and you'll take the piece of paper to the local pathologist and they'll have a blood test, and if that's the answer, great. If it fits the doctor's idea, great. If not, you go back and you'll start another test. I'm not doing well with this technology as much. So in life, some conditions are genetic. We're learning that genetics is contributing to more and more conditions that we never thought were genetic in basis, and you've heard about that from some of the early speakers. And we all have 20,000 genes, and in the average one of you, not all of those genes are actually working. And we now know that somewhere between half a dozen and a dozen of them are faulty. They're not working. But the reason that you've reached this stage in life and that you're here listening to me today is because you happen to have a spare copy of each gene on the other chromosome. As you've heard from Marie's talk earlier, we all have 2 copies of everything. And so the faulty gene may be recessive, you may have another copy on the other chromosome. Oh, I'm getting the wrong way now. Okay, so if you'd like to try and sort out which of these conditions might be genetic, your doctor will send a specimen to the laboratory, to a genetic laboratory, and we'll approach it in one of several ways. We used to do a genetic test, and as technology has advanced and enabled us to do things, we can now do increasingly complex types of genetic tests. And I'll just walk you through each of these different types: single gene test, the gene panel, the whole exome sequencing, and the very exciting whole genome sequencing. So with a single gene test, 20,000 genes, your condition, your symptom signs can be explained, we think, by just one condition, and we happen to have a genetic test that does that. Examples might be cystic fibrosis, which happens to be a very common condition within our community. About 1 in every 20 to 25 of you are carrying a fault in the cystic fibrosis gene, and about 1 in every 2,500 children born in New South Wales will have this condition, cystic fibrosis. Tay-Sachs disease, even more severe condition that affects in our community mainly people from the Jewish community who come from Eastern Europe. Hemochromatosis, a disorder of iron absorption. We have a number of these different conditions that are very common within our communities, and we have them because our ancestors have come from a part of the world where these gene faults were common. Now, testing for single genes is reliable, it's simple, it's relatively straightforward, it's relatively cheap, somewhere around the $100 or $200 range, and it's a bit hit and miss. If you come up with the answer, you've got your diagnosis. If you haven't, your doctor goes back and comes up with another hypothesis and requests another test. And that's where we were until about 10 years ago. Well, rather than going for just one gene at a time, why don't we gather together similar genes that might cause the same condition? For example, you might have aches and pains in your muscles. So let's look at all the genes that can cause aches and pains in your muscles. Or you might have a weak heart. There's a panel of 200 or 300 genes that we now know can cause a weakly beating heart. And instead of doing the test one by one by one at $100, $200 a time, we'll gather all those genes together and we'll do in one go all 200 or 300 genes. That's a little more expensive to do, but you get your answer for the whole spectrum in one go. Now, that was a huge step forward, and that's roughly about the state of the art, as of, say, last year or this year. The advantages are terrific to pull them all together. It's very exciting, very powerful. The disadvantage means I share the excitement of my colleagues when they make a new discovery. I then groan with the thought I'm going to have to redesign my gene panel because this takes over a year of work to revalidate it, and I then can't offer the test for another year till I've incorporated that gene, by which time there'll be another discovery and my gene panel will be out of date again. So it's a process, it's the best we have, but it's imperfect. So let's take our reasoning process one step further. Let's look at all possible genes. You've heard from early speakers, we have 20,000 genes, and if we just focus on the genes that do something, let's not worry about the controlling genes at the moment, there's 20,000 genes. At the limits of technology, we could do a gene panel for all 20,000 genes in one go, and that's called a whole exome sequencing. We're going to look at all the genes in the expressed region of the genome, the exome. Very high degree of expertise to do it, laboratory expertise, computational expertise, and medical interpretation expertise, but it's possible. We find, to our delight, that about a quarter to a third of conditions that were previously thought to be undiagnosable turn out to be able to be diagnosed with a whole exome, because you look at every possible gene and you think, oh, it didn't even occur to me, it's a genetic condition. And again, we had examples earlier where you can either approach it through a hypothesis or through this very powerful shotgun approach where you look at all possible genes. And some examples are some of the immunodeficiencies are being sorted out this way, and also some of the very serious forms of epilepsy are beginning to yield to this approach. Well, we can take it one step further. Instead of looking just at the expressed genes, let's look at all possible genes, the entire genome, which is 100 times bigger, 100 times more complex. Interestingly, it may actually do an exome better than an exome test does because of certain technical reasons it avoids bias. It includes the spaces between the genes and the controlling genes. The problem with that is if an exome was harder, the whole genome sequence is even harder, 100 times harder, more than 100. The combinatorial possibilities are huge. So it's a very, 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 very complex task. And at the moment, there's only 3 places in the world that can do it, of which the Garvan is going to be one of them. And that's the reason that I'm here. The New South Wales Health System thinks this is so important and so exciting that they've lent to me. I'm a hard gun, so to speak. I've been lent to the Garvan for a year to help the Garvan rapidly accelerate the process of medical accreditation. So by the end of the year, the testing will become available to the New South Wales Health System. So there's so much information involved. Technology and understanding is driving it. That only 15 years ago, the first human genome was sequenced. It was 3 billion bases. It's a coincidence it was about $3 billion it cost to do that. Today, we're somewhere around the $1,000 to $3,000 range, $1,000 in the chemicals plus a bit of interpretation. And the cost will keep on dropping as it goes. So that's where it's at the moment. The laboratories, this is the photograph of actually the laboratory next door in the Kinghorn Centre where it's done. This is the previous generation. The current one looks like that. It's just on steroids, 10-fold bigger. The machine is actually, as we're speaking now, being tested and commissioned and validated for introduction later this year. And the way we apply to medicine is we start with the 3 billion possible letters of the alphabet, 3 letters of the DNA alphabet, and Mary mentioned about 1 in every 1,000 position varies between you and the person next to you. That means you've got 3 million variants from the hypothetical average human. And with computerized techniques, we rapidly go from 3 million down to the ones in the gene that are going to make a difference, the ones that are not actually common within the community. And that's the end of the computing part. And for the last bit, we sort through those one by hand, and we try and sieve it down to the last dozen, the last handful, the last 1 or 2 that may actually explain what condition the patient has. So this process here is about a week or 2, and that process can take up to 6 weeks as we go through and we hand curate every single result till finally we issue diagnosis. And so about 1 in every 3 cases that comes up, if you pick them carefully, you can come up with a brand new diagnosis that medicine and science has never seen before. So let me give you an example. This is a real-life example from the Children's Hospital, one of the cases that Chris and we were collaborating on, or our colleagues were collaborating. an 11-year-old girl. She's had a long and stormy time through life with recurrent infections. She has 2 healthy parents, 2 healthy brothers and sisters. She's the 3rd in the family, and unfortunately, her lot in life was she's had a very sickly childhood. Very serious infections. She's had abscesses growing inside her eye. When they've been cultured, they look like tuberculosis, but TB doesn't grow in the eye. She's been treated with all sorts of very nasty antibiotics. And so the thought was, well, has she got some form of immune deficiency that is preventing her from fighting these bacteria? Except there isn't actually a condition that fits. There's one that's come close. So, armed with a bit of a clue, they sent her DNA off to America, who sequenced all of the known genes that could cause anything remotely like this, and they didn't strike a hit. So she had some probable immune deficiency, we don't know what it was, and she commenced treatment with interferon, which is very expensive, about $250,000 is the cost of the treatment, and it has a lot of side effects. So the thought was, well, can we help this family? Can we help this child? And yes, so we did. So we took the DNA from the healthy dad, from the healthy mum, and then from the affected child. And by knowing what variants were present in both parents, it greatly simplifies the task of looking for what variants are in the child, because many of the variants you see, you know either mum or dad's got them and they're not affected. You can cross them off the list. They're probably not going to be the cause of the disease. And we discovered that by looking at this dad, mum and child, the so-called trio, a brand new mutation had occurred in the child that wasn't present in the mum or dad, and it was in a gene that was actually very relevant to the immune system. So then, knowing where to look, we went back and confirmed it, did some studies to show that it probably was the cause, and the diagnosis was made that we can now help that child, stop the incorrect treatment, save the family and the health system a lot of money, and help the child with a more appropriate treatment. So the pros and cons of the whole genome sequencing, which will soon be available here, incredibly powerful. It finds everything. Best value you get for the cost of one test, you get everything. Lowest cost per gene, and it's clearly the way of the future. As the costs drop, this is going to become part of mainstream medicine. The cons, well, it requires enormous expertise in science, in information technology, in medicine. It's expensive if you don't need everything. If one gene's enough, stick with the one gene, but increasingly as we look for more complex diseases, we'll be looking for packages. And the other interesting issue is the issue of incidental findings. This is important. Let me explain what incidental findings. You look for one thing, but in the process you discover another thing. You have a chest x-ray because you've got a cold and you discover there's something here that might be cancer. You have, in the case of your DNA, you look for your genome looking for one condition and you find something that you might not necessarily want to know about. You might find that you've got an inherited susceptibility for certain diseases. You might find that you're going to have a neurodegenerative condition that will affect your intellect as you get older. So it's very important that we know what the patient's wishes are, because once we find it, we can't tell a fib in the report, we have to report it. But if we know in advance what the patient wishes to do, we can adjust the report, so we only report back to your doctor what we know you've given consent to do and respect your wishes and your privacy. So the way this will be applied for medicine in the very near future, perhaps even as soon as next year, is when you go to the doctor, not feeling well, the doctor will still be there and they'll still ask their questions of you and they'll still listen to you and they'll still try and form the hypothesis, but instead of then coming up with an idea and they test, and another idea and they test, and another idea and they test, the very first thing that happens is they might go and do a DNA test, whole genome. That's not next year, but shortly after, whole genome. And armed with the whole genome, they'll look with the computer and they'll come up with what conditions you might have. And then from that, they'll then compare to what your actual symptoms are, and they'll say, ah, you've got this. And you don't have to go through the long, complex tests, hit and miss tests, all the other tests. But to do that, we need to know what the patient's wishes are. So, we are really at the dawn of a very exciting time in medicine. The impact of this will be equal, as you saw in David's earlier slides this morning. It's going to write the history of the future of medicine now. We will, in some ways, it won't be the actual treatment that will be important, it'll be the understanding of disease processes. It will unlock our understanding of what causes diseases. And from that, as we've heard, we will be designing newer and better and far less toxic treatments that will come from it. The costs are rapidly falling. Yes, it's $1,000 to $3,000 now. It'll keep on halving. So very soon, I've been to a conference, I haven't got time to include the slide here, where they're actually giving them out as a freebie to conference delegates. Would you like to have your genome sequenced? Not everyone wants to know. I carry my genome here. I was intellectual exercise. I had my genome done, and I did it because I want to know if I'm advising patients. I can't counsel them unless I know the thoughts that I've had when I went through. And we discussed in our family, and my wife said, under no circumstances am I to tell her anything about my genome. She doesn't want to know any of the findings. And my daughter said, under no circumstances am I to withhold any information. We are all different. You need to know your views, so we have some very important ethical issues there and some very exciting and powerful new technologies that will become available through the Garden from Eden.